when we think about that vision of building virtual cells, there's a lot of unknowns and we're purely data limited. But I am myself very convinced that both protein data and image data, so spatial proteomics data, will be key in order to be able to understand cells as the functional, complex systems they are. Proteins are really the building blocks of cells. They are encoded by genes, but it's really the proteins that build up the structures in the cell and they perform most of the functions in the cell. Proteins are also the most common drug targets, for example. So when we want to understand how cells function, we need to know where proteins are and understand what functions they do, for example. So it's vital for everything from basic biology to drug discovery. And spatial proteomics is a group of technologies that are really aimed to not only measuring the abundance of a protein, but to really understand where are they. If you think back, I guess more than 20 years now of the Genome Project, at that point we knew that these are the genes that are encoding human biology. We did not know what the corresponding proteins were doing, not all of them. So knowing where a protein is located gives us very good clues about its function. If we know that this protein is expressed in hepatocytes and it's located to the Golgi apparatus, that gives us some clues about what processes that protein might be involved in. So that is the reason that we want to map protein spatial distribution across biological systems. The example we're most proud of is, of course, the Human Protein Atlas, which is a tour de force of spatial proteomics, if you put it that way, where we've generated antibodies for the majority of all human proteins and then mapped where they are located spatially within the cells and across most cell types of the human body. Because we believe that with open science and open atlases, we can accelerate science better together. And we were fortunate enough to have funding from the Lundberg Foundation so that we've been able to build the Human Protein Atlas and we can produce data at scale, and we want to share it with the rest of the world because there's so much information captured in there that can be interpreted through many different lenses depending on which research you're doing. And of course, we want to share that with everyone. So it's really a type of cartography, I guess, of biology that would be very hard for an individual lab to do, but a big program like the Human Protein Atlas can do it, and we're happy to share the results with the rest of the world. I think it's a natural follow-up on spatial transcriptomics being method of the year a couple of years back. It's harder to measure proteins than it is to measure RNA. We don't have a PCR for proteins, for example. We're learning how to interpret the data better and better, and also using AI to interpret this data and build models up from this data. So I'm not surprised that it's the method of the year, because we've seen in the past 5 years just a multitude of spatial proteomics methods being developed that measure slightly different things, and we've seen beautiful examples of how spatial transcriptomics and spatial proteomics data is being integrated, and we can really see how spatial proteomics data will also be a very important data source for future, I guess, cell mapping efforts and virtual cell modeling and such efforts. There are many different developments that I would want to see and that many of us wants to see, and I think it's a whole suite of technologies. It's hard to say that there is one next thing. I think that analysis is always a bottleneck. With AI, we're going to get better and better and better at analyzing the data and really modeling off from that data. So I think one important next step will be that we start to build spatial models of cells from spatial proteomics data. Another development is that I think our assays will be measuring more and more proteins, so better and better coverage of the proteome in spatially resolved methods. I also think that we'll see more advanced spatial methods that, for example, doesn't just measure the location of the proteins, but maybe interactions between specific proteins in a multiplex setting. Using the images we've generated in the Human Protein Atlas, we want to build a spatial proteomic model of a human cell. So that is something that we're working hard on now. And we also want to understand dynamic spatial regulation of proteins. So proteins that tend to translocate, move from different parts of the cell, from one part to another, as the cell is kind of dynamically adjusting its function. So in a non-genetic way, fine-tuning its function to its environment. Our lab is fully focused on spatial proteomics, so we'll continue to do spatial proteomics. We are moving to organoids instead of cell lines. We're working more in 3D instead of 2D, and we are working a lot with AI to harvest all the rich data and images that we have in spatial proteomics, and also want to integrate it with other types of spatial proteomics data, like protein-protein interaction data. And ultimately, we want to build that spatial model of a human cell and its proteome. Yes. Right now, there's a lot of hype around trying to kind of see that vision of a virtual cell. So can we finally, with the help of AI and the large data sets that have been generated in biology recently, start to build virtual models of human cells so that we can simulate cell behavior and predict function from cells? So ultimately, you can kind of envision that you can make a clone of my cells if I need to get a treatment for a disease or something, and decide which treatment I should get, for example. And while that is still a very much science fiction, I guess, vision, the developments that we've seen in large language models and also vision transformer, for example, positions us in a new place to really think about building integrated models of cells that are learned from large corpuses of data. And I think this is a very exciting time for that. When we think about that vision of building virtual cells, there's a lot of unknowns, and we're purely data limited. But I am myself very convinced that both protein data and image data, so spatial proteomics data, will be key in order to be able to understand cells as the functional, complex systems they are. Because a cell is so much bigger than a single protein, and if we want to understand and simulate cell function, we're going to have to assemble a cell, or whatever you want to call it. And to do that, we need to know where the proteins are located in a cell. We need to know how cells are arranged in relation to each other, and a good way of measuring this is through spatial proteomics. So I think that spatial proteomics data will be key for any future virtual cell modeling efforts, and I think that we have to really think about how we can scale up the data generation to make it both proteome-wide or closer to proteome-wide, but also to be able to just generate more data across more samples and more cell types. We're talking like a decade or something. It's very hard to speculate when we'll see such functional models. I think we're still very data limited in terms of building such models, but I think there's a different question that can be asked. When can we design models that are of utility to people that are in the lab? So, for example, I think that a couple of years from now we'll have models so that we can, in our lab, simulate what stainings and microscopy images, spatial proteomics data will look like in a certain sample before we actually do the experiment, and that those models will help us to design the experiments better. And then we're not as dependent on the models being super accurate on everything, as long as they're good enough to help us really design our experiments. So I think that's where we're going to see the first implementation of such models, and that might be a generative image model or a generative sequence model, for example. If you think of how you use ChatGPT now, it knows everything on the internet, basically. So if you can use models of cells to design, you have a question, I want to answer this research question. Should I look at this set of proteins or this set of proteins? Which tissue should I look in? How should I design my experiments? So there might be a lot of tools that can really help us to do that quicker. Whereas the full virtual cell models, I think that will take a longer time. There's a lot of challenges. I still think we're data limited, and that will take a while before we figure out what new type of data we need to generate and which cell type we need to measure it on and how we share that data and integrate the data. But a problem that I think all of us in academia have is also that we're compute limited. So this is really, it's a huge, building virtual cells is a huge project that needs collaboration across the world, across sectors, across many labs. So it's nothing that one lab can do by itself, but it's a collective effort. But that's why we have a more narrow focus. So we want to build that first image-based spatial model of the proteins in a cell, which is far from that virtual cell where you can simulate functions of a cell in any given condition, but it is a sliver that we can work towards. We know how to fold proteins now with AI, as been demonstrated brilliantly by many people, but a cell is so much larger than a protein, and in order to really ever model a cell, a challenge would be to be able to learn across scales, to be able to both understand things at the molecular level, but also to simulate function of the entire cell. And I think spatial proteomics is one of those technologies that actually maps to that entire spectrum of scales. So that's why I think it will be useful. So SubCell is a suite of vision transformer models trained to be able to do many different tasks related to microscopy images of cells. So images are incredibly rich in information, and they have more information than we can even interpret with our eyes. So this is a suite of models that we trained together with the Chan-Zuckerberg Initiative, and they are really generally applicable, highly performant models that can interpret patterns in cells. It can be used to understand the phenotype, predict mechanism of action, if it's drug screening data, for example. So it's really one of these, I think, next generation tools that we're seeing, how AI is really helping microscopy research. We can turn basically any microscopy images that we've tested so far into quantitative we can get a quantitative information and quantitative embedding of cells from microscope images. And with that embedding, we can, for example, integrate it with other types of data or build a model of the proteome of the cell, which we did in that paper. This model is great because it allows us to interpret spatial distribution of proteins in single cells from microscope images. And that's great. What we really want to build is a generative image model, an image model where we can, almost like in a microscope, see all human proteins in one image, more or less. And that is a different task. And there we also need very high spatial precision in that generative task, and that is something that is hard to achieve at the moment. So far, we've demonstrated its usability in several different tasks, but one very exciting aspect is that we built the first data-driven, model of the proteome in a cell. We used the embeddings that we get from SubCell and built this hierarchy of functional systems in the cell. So the cell, and then we have the nucleus and the cytoplasm, and then it kind of gradually factors down into smaller and smaller organelles and structures. And in the end, we get to these smaller systems that have associations to certain biological functions, way finer than we could ever annotate with our eyes. So you can think of it as a kind of an equivalent of the gene ontology cellular component, but learned directly from data, no human intervention. It's learned directly from microscopy images, and it shows us what the proteome architecture of a cell looks like. Yeah, in a kind of condensed format, like a map. It's a kind of simplistic approach, but it still shows that you can really embed proteome-wide information into a format so that we can aggregate and build this map of 14,000 human proteins into a simple hierarchical map that shows which proteins are in close proximity to other proteins. And this map in itself might be very valuable for integration with other models. For example, if you have a language model that has learned to encode single-cell sequencing data, maybe you want to condition that model on the proteome architecture, for example, so that the model learns causal reasoning better across biological pathways. I think that this proteome graph that we built thanks to SubCell, that graph can be used for many different things. It can be used for all the different tasks that gene ontology is used for today. But it is truly unbiased, data-driven, and I think we'll see that it will be integrated into many other types of sequence-based language models as well, as a kind of structure for learning how genes relate to each other. Yes, we have. Yes, I would love to talk about that. So one of the recent additions that we've done to the Human Protein Atlas is that we've added a section on primary cilia. And this primary cilia is one of the organelles that we have never covered in the protein atlas before, because it's not present in the cell types we had previously measured. So primary cilia are very fascinating organelles. They're like an antenna that certain cells have, and on that antenna you have a lot of receptors. So basically this antenna is sensing the environment and signals, and that it can relay to the cell. And the primary cilium is incredibly important for development, for example. So most things that we know about the primary cilia are through diseases called ciliopathies. When proteins in the primary cilia are dysfunctional for one way or another, you get ciliopathy diseases. So we know that there's been a great interest, and people in the cilia field have also often asked us in the protein atlas to generate a cilia atlas. So we're very happy that we finally did it. But it's been hard because the cilia is so small and fragile. So this is the first time we actually had to map cells in 3D instead of 2D in the protein atlas, and it's been a challenge. But in the end, we released a very nice version of that atlas, and we found many new cilia proteins, and we also, even together with collaborators at the Karolinska, identified a potential mutation in a new cilia protein that might lead to ciliopathy. So it's been a very interesting study, and I hope that cilia researchers out there will use it. And that is actually, cilia is a great example of why spatial proteomics is important. So if we look at all organelles, the cilia is the one organelle with the highest heterogeneity in terms of its protein composition. So it's highly tuning its proteome composition to the cell type it's on. But also within a cell type, we see a lot of heterogeneity. So it seems like the antenna is really fine-tuning its ability to receive signal based on the state that the current cell is in, to an extent that we don't see for any other organelles. So I think that is very fascinating. But most of the proteins that are in the primary cilium, they are also expressed elsewhere in the cell. So this is nothing that you can get by gene expression, because the gene will be expressed in many proteins, but then in many cells, but in some cells, the cell will choose to put this receptor, spatially we localize receptors to the primary cilium, and that's when they can receive these signals. So it's a cool process that is highly spatio-temporally regulated, but incredibly important for cellular function. So it's a great example for why spatio-temporal distribution of proteins matter. It is a way that cells control their function.